Now we'll discuss about stability study. Stability study is very important, crucial and very interesting topic of any pharmaceutical sector. Even it is a vital part for product development also. You can determine shelf life of any medicine from this study and many more things. See, there are several documents, books and so many guidelines regarding stability. If you want, you can see or you can read WHO guideline for reference. Even in 21 CFR part 211.166, we get some information about this stability study. But yes, mostly we are following very popular, very common ICH guideline for particularly this stability study. Remember, last we had discussed about ICH guideline, multidisciplinary, then efficacy, safety, then quality, right? So in here, quality part, Q1 chapter is specially for stability study. Now let's have a look what is inside in the Q1. Here, Q1 consists so many subparts. Q1A, Q1B, Q1C, D, Q1E, Q1F. So Q1A, stability testing of new drug substance and new drug product. Then Q1B, photostability testing of new drug substance and product. Here, this Q1B guideline explain us how do we perform photostability study. Here you can see a simple flowchart which nicely describes the procedure stepwise. Just recall the force degradation study in method validation part, where we get to know that pharmaceutical product exposed at light having not less than 1.2 million lux hour or not less than 200 watt per square meter for check the impact on quality. Just similar in here also. Move to the next guideline, Q1C, stability testing of new dosage form. Followed by guideline Q1D, bracketing and matrix design for stability testing of new drug substance and product. Here, you can prepare a design protocol in such a way so that it can reduce your analysis schedule, which neither impact your data quality nor a significant change in your predicted shelf life. Next guideline is Q1E, evaluation of stability data. Believe me, I think it is most important part in stability study. This guideline describe us how do you prepare analytical data format for a good presentation, and you can know how and when extrapolation can be considered. And then last guideline over here, Q1F, stability data package for registration of climatic zone 3 and zone 4. So now you can imagine how important the content Q1 in ICH, or you can say stability study in pharmaceutical industry. If not, I will share you one more things which will definitely help you to realize the aim of stability study. See the general principle. I think it is present in every guideline. The purpose of stability testing is to provide evidence on how the quality of API or finished pharmaceutical product varies with time under the influence of variety of environmental factors such as temperature, light, humidity, and to establish a retest period for the drug substances or shelf life for a drug product and the recommended storage condition. So from this sentence, one thing is clear that quality of pharmaceutical product depends on time and the environmental factor. Apart from this factor, some other factor which more or less influence the product quality, like interaction between the API and excipient, manufacturing process followed, type of dosage form, container closure system during packaging. These are also impact your product quality. Impact your product quality means product may undergo change their appearance, consistency, content of uniformity, particle size, shape, even pH, moisture content, etc. These changes may occur due to product's molecule internal vibration, abrasion, or you can say temperature fluctuation like freezing, thawing, searing, etc. You observe almost every product are temperature sensitive. There are two potential area of temperature sensitivity: temperature above and the temperature below, in the range where the product has been shown to keep their unique property until a certain period. At very low temperature, pharmaceutical products are usually damaged by losing their therapeutic property when the product has been frozen, and their active ingredient structure may have irreversibly changed. On the other hand, products behavior is often different at very high temperature also. Pharmaceutical products gradually lose their potency in API as well as in excipient. Molecules are degraded through transformation here. Degradation reaction like racemization, oxidation, reduction, hydrolysis, solubilisis may occur at this time. Naturally, depreciate formulation property, significantly reduction excipient activity like antimicrobial preservative action and antioxidant, etc. Same like temperature, products impacted by high humidity also. Here product absorb extra moisture content resulting degradation. Even some product get toxic due to absorption of moisture content. Product turns sticky or crumble in nature, so impact on hardness, then chances to grow microorganism at this stage. So till now, whatever we have seen, it made us to realize that degradation rate of pharmaceutical product is much more at high temperature and high humidity than the ambient temperature or ambient atmosphere. On this basis, we keep our product at stress condition, usually 40 degrees centigrade, 75% relative humidity, to check the impact on the quality of the product within a short period. See, depending on nature of the product, elevated stress condition is different. Normally, we keep our product for only 6 months. And analysis frequency minimum 0 month, 3 month and 6 month. If you want to increase your testing schedule, it should be mentioned in your stability protocol. Okay, I forgot to say that this type of stability study is called accelerated stability study. This study based on Arrhenius equation, or you can say modified Arrhenius equation, which describe the relationship between the storage condition and the degradation rate of the product for prediction of shelf life. Well, shelf life commonly estimated using two type of stability study. One is from accelerated stability study, another one real time, or you can say long term stability study. In real time test or long term study, product is stored at recommended storage condition. See here different storage condition and its activity monitor until it fails the specification, or you see here. For long-term studies, the frequency of testing should be sufficient to establish the stability profile of the API, or you can say formulation product. For API with the proposed retest period or shelf life of at least 12 months, the frequency of testing at the long-term storage condition should normally be every 3 months over the first year, every 6 months over the second year, and annually thereafter the proposed retest period or shelf life. I'm again trying to say that your analysis frequency and then in which storage condition you keep your samples should be mentioned in your protocol. Not only storage condition, your sample consumption record, analysis data and its evaluation summary sheet, trend, etc. to be mentioned in your protocol. And how to prepare your summary sheet, trend chart should be written down in your stability SOP. And how to arrange your stability chamber with sample in sequentially manner, then labeling for better identification should be mentioned in your SOP. How to maintain your stability chamber to be described with proper way in your SOP and written down the solution if any incident occur, like temperature, humidity goes out of limit, then instrument breakdown, even abnormal result, or you can say significant change observed during analysis, means full of information. Remember, make your SOP clear, precise and understandable. Thank you. So make your SOP with your voice.